Let's take a look at excitation-contraction coupling. This slide here summarizes everything. It's just really hard to see everything all at once, and so you can go back to this slide and look at the different steps. So we're gonna do first the part A, the initiation of the muscle action potential. In other words, how does the signal go from the motor neuron down to the muscle? So the action potential is actually coming down, racing down this motor neuron, and it's arriving at the axon terminal. We learned that at the axon terminal, there's calcium channels, and they're going to open up, and this action potential is racing down here. So it's action potential arriving here. Calcium influx causes these vesicles that contain acetylcholine to fuse with the presynaptic membrane right here. Then the acetylcholine that's in here, the little red dots, it's going to be released from these synaptic vesicles. It's going to diffuse across the synaptic cleft, and it will bind to acetylcholine receptors. And these happen to be nicotinic type of acetylcholine receptors. They're ion channels, and in response to the acetylcholine binding to the acetylcholine receptors here, these ion channels will open up, and that causes a influx of positive ions into the muscle cell right here. So this is your cation channels, and a lot of positive charges are coming in when these channels are open, and that causes a depolarization of the muscle membrane right here, the sarcolemma, and that causes then an action potential to be traveling in both directions as a depolarization wave here. As you can see, that this action potential is now racing down both sides along the surface here of the muscle membrane, the sarcolemma, and it's going down here. Once it hits a T-tubule, it will be going down deep inside of the muscle cell. So here on the next slide, you can see this action potential that's now traveling on the surface of the muscle fiber, and it's going down the T-tubule right here, and there it will hit two calcium channels that are linked to one another. First, the DHP channel, the DHP receptor it's called. That one is a voltage gated ion channel, and it's linked, physically linked. So you see this little tethering thing, this little spring thing. It's linked to this ryanodin receptor. That is a calcium channel that opens up, and it will let the calcium that's stored in the sarcoplasmic reticulum, it will cause it to come out. So let's take a look at this junction right here. That's a very interesting part right here. So we have the DHP receptor right here. That's a voltage gated channel. It opens up, and as it opens up, it will sort of snap open this gate right here, if you can see that. So this gate is now open right here, and the calcium can now come out. And we learned earlier that the calcium then will bind to troponin, and that causes the troponin to change shape, and that makes the tropomyosin come off of the actin, and there we go. Now the myosin heads can latch on to actin, it can do that pivoting thing and produce the power stroke, and that will shorten the sarcomeres, and that is seen as a muscle contraction. So here, up closer, one more time, here's the DHP receptor, and then the ryanodin receptor. The ryanodin receptor this is sort of a physical link right here between these two channels. When this one changes shape because of the depolarization wave coming through here, then it yanks open the gate of this calcium channel, the ryanodin receptor, and the calcium will come out, as you can see right here. Then the calcium will bind to troponin, so the green calcium will bind to the troponin, and then the troponin changes shape, and the tropomyosin comes off of the actin. So here, continuing sort of step by step. Here again, the calcium is now binding to the troponin and changes the shape, and now we're moving on to once the muscle has produced its contraction. Now we need to pump the calcium back into the stores, and that is a ATPase. It's a pump that operates under expenditure of energy. It will pump the calcium back into the stores right here. And once the calcium concentration is low, it will be released from the troponin that causes the troponin to change back to its original shape, and it's also moving the tropomyosin back into the original shape, and that covers up the binding site for myosin on actin, and that will lead ultimately to relaxation. So here up close, this calcium ATPase will pump the calcium back once the muscle has contracted. Here you can see the gate is now closed again, and the calcium is being pumped back into the sarcoplasmic reticulum, and that means the calcium will be released from the troponin, and troponin will change back to its original shape, and it will move the tropomyosin back to its original position, and that means that actin and myosin can no longer interact, and that means the muscle will relax. Now, if we were to try to record what's going on in the muscle by sticking a electrode into the muscle, and then stimulating it with an electropulse to simulate the arrival of an action potential at the neuromuscular junction, and then recording the subsequent contraction of the muscle. So that would be a muscle twitch recording. And if you did the lab, I think it was 2 weeks ago, if you did that lab and watched the videos and did the lab style exercises, then you would have seen a single muscle twitch recording. That was the big thing for that. And you would have also seen it as a short latent period. And that latent period is the time between the stimulation of the muscle to when it physically actually contracts. And as you know now, there's a lot that needs to happen, because the DHP receptor needs to yank open the ryanodin receptor, the calcium needs to come out, it needs to bind to troponin, and troponin, tropomyosin need to move away from the actin, and then the actin and myosin can interact with one another. So for all of that to happen, you need to allow a little bit of time, and it's generally about 10 to 20 milliseconds, depending on how responsive that particular muscle is. But if you would have recorded this yourself, you should have seen something like this. This is pretty good down here. So right here, let's focus on this. So you would have stimulated the muscle here, and then you have this latent period of we usually record it at about 20 milliseconds when we do it in the lab as a simulation. Also, we used to do this on real frog gastrocnemius muscle, as you had in one of the YouTube video clips for that lab. That's when we measured around 15 to 20 milliseconds for latent period. And then once the actin and myosin are cross-bridge cycling, that means we're actually building up tension all the way to a peak. And so this is the beginning of the contraction to the peak. That's known as the contraction phase, and it's about 50, 60 milliseconds long, depending on how tired and responsive your muscle is. And then the relaxation phase, actually, most of the time what we find is it kind of trickles out a little bit more. It's not quite as symmetrical as they want to make it seem here, but nevertheless, so here the relaxation phase is from the top of the contraction, so the maximum tension, all the way back down to baseline. And what we have found is usually it's about 100 milliseconds or so. Here they have the entire contraction and relaxation at about 100 milliseconds. Well, anyway, it depends a little bit how you're recording this, and the timing of all of this is not as important as understanding the stages here. So superimposed up here, we have the action potential, and so that is the beginning of the depolarization here. And so then we have in muscle, the muscle responds to the stimulation with a latent period where we need to do the whole calcium story. And then we do contraction and then relaxation. Now let me say a brief word about phosphocreatine. You should think of it as a secondary fast source of energy. So the muscle has some stored ATP to do all this work of these cross-bridge cycling. And as you know, every cycle of the myosin heads latching on and detaching and recharging costs 1 ATP, and that's per myosin head. So each sarcomere needs a lot of ATP, and then of course the muscle fiber as a whole needs a ton of energy. So there's enough energy, ATP, immediate energy, for about 20-30 seconds worth of work, and then you need to start getting into your reserves if the muscle is supposed to continue to work. And your secondary source of energy is phosphocreatine, It's an organic molecule that has this phosphocreatine. And so it's basically, you can use it to recharge your ATP quickly. And so you can use the phosphate from the phosphocreatine to slap it back onto ADP, and that makes you some brand new ATP that you can use. So you can use the phospho from the phosphocreatine plus the ADP, and you make brand new ATP really, really fast. Once that source is used up, then you're going to have to churn out more ATP by starting your aerobic metabolism engines and producing ATP. Muscle will fatigue eventually, and that's mostly due to depletion of these energy sources, and sometimes also lack of oxygen delivery that then causes buildup of lactic acid, and that gets you even faster into fatigue. So there's no particular big thing that we need on this slide, so just for your information, you can take a look at that. So here's some causes for muscle fatigue during exercise. It's usually just running out of energy sources, immediate energy sources, and then a lot of times it's also the buildup of lactic acid that plays a role. So here are some sort of levels that you can see between rest and fatigue. So taking a look at the ATP, the lactic acid buildup, phosphocreatine versus the creatine level. So you can take a look at it. It's not particularly super important for us. And then here, moving on to fast twitch versus slow twitch fiber. The slow twitch fibers are your endurance fibers. So that will be, they rely on oxidative phosphorylation. That is your deep red meat, your endurance muscles that you use for maintaining your posture, or think of the ultimate endurance muscle, your heart. Those, it's predominantly slow twitch fibers because they are doing the marathon work. Think about how much vacation do you give your heart. So you're expecting your heart to work 24/7, 365, no breaks, no vacation. So you better make sure that your heart gets everything that it needs in terms of oxygen, nutrition, because you don't allow any breaks. And your heart truly is the ultimate endurance muscle. And in opposition to that would be fast twitch fibers. Those are, well, like the name says, they're fast, but they fatigue pretty fast too. Fast to go and fast to fatigue. They are mostly glycolytic. They rely mostly on anaerobic glycolysis for a little bit of quick energy, but they fatigue really fast. The fast twitch fibers, those are your thicker fibers, and you kind of can tell when somebody's more of a sprinter type and doesn't really have much endurance. Those are people with the bigger, bulkier muscle, versus the marathon runner isn't super bulky. Those are the skinny fibers that are mostly oxidative and slow twitch endurance fibers. But here is sort of histology slide that shows you different fiber types. So there's not super whole lot of that you can actually distinguish here, but you can see that the fast twitch fibers, they're much bigger in diameter. These are huge fibers versus the skinnier, smaller fibers. Those are your oxidative fibers that are endurance muscle. So by the sheer size of a muscle, you can tell, the bigger, bulkier muscle, that's, strength, but not much endurance. Your weightlifter, for example. So here's a comparison of the characteristics of different muscle fibers. You can take a look at how fast they process ATP, the ATPase activity, the speed of the muscle itself, and some of the stuff that we just talked about. Postural muscles would be endurance muscles, of course. Your heart's endurance. And then the fast ones, those are the ones for quick jumping or just for quick movements. Strength movements, actually, that is. And here, a quick look at a length-tension relationship. The point of this is basically, you can see the best tension, the maximum strength of these sarcomeres is achieved when they're not overstretched like they would be here, or completely crunched together like you would see over here. So you want to have some space so that you can develop maximum tension, and that's pretty much the point of this slide here, to show that you don't want to have too much overlap to begin with. You don't want to have fully stretched out without kind of sarcomeres like you had here. So you want to have sort of in between to have a nice ability to develop strength and tension.